Hello everyone, my name is Kayla Aguilar and I will be teaching you all about mononucleosis. Mononucleosis, also known as mono or the kissing disease, is an infectious disease caused by the Epstein-Barr virus. This disease is prominent in adolescents, and roughly more than 90% of adults in the U.S. have been exposed to it by the time they are 30 years old. It affects the whole body, from the respiratory system to the digestive system, muscular system, integumentary system, and the lymphatic system. I'll have more details about this later. And a fun fact for you, mono is technically herpes. You heard that right, herpes. I'll continue this in the next slide. There are many different pathogens that can lead to mono. However, the Epstein-Barr virus, EBV, from here on, is more prominent as it is one of the main causes, and it's what I'll be focusing on today. So mono is caused by a virus. What does that really mean? It means it has its own classification. Here it is known as the human herpes virus, HHV-4 to be exact. This means it's one of the more successful viruses, as it could easily adapt to its host, alternate between replication and its latent infection. To classify it even more, EBV is a gamma herpesvirus. It's different from alpha and beta, as it is able to develop lymphomas. To classify it even more, it is a prototype of the Lymphocryptoviridae generation. In the upper right-hand corner, you could see a nice 2D cartoon drawing of the EBV structure. You can notice its viral characteristic of the core being surrounded by a nucleocapsid encapsulating the DNA genome inside. Though it looks 2D, EBV is actually toroid shape. Toroid shape is what you see in the bottom right-hand corner. The toroid shape allows the envelope proteins to increase its surface area in order for it to easily latch onto other cells when replicating. With its unique shape, its DNA is accessible and causes a difference in sequence, which leads to EBV1 and EBV2. Its genome, right in the middle, is linear and double-stranded, having more than 85 genes. Having 85 plus genes divides EBV into latent and lytic genes. What does latent and lytic mean? To fully understand them, let's break down how it's divided. For its DNA genome, you would need an open reading frame. An open reading frame is a portion of DNA which is to be read for coding. For EBV's case, this open reading frame divides the coding into latent and lytic. These two subdivisions are significant when infecting and replicating in a host. There are plenty of details to how and what each division does, but I'll give you the gist. Latent has a significant role during the infection state, as its coding allows it to maintain plasmid, replicate DNA, and regulate transcript. Lytic becomes important during persistent infection, as its genes allows it to be similar to the human proteins and allows it to integrate DNA replication and amplifies the virus. This becomes important when we talk about the EBV's life cycle. Speaking of life cycle, since I mentioned some parts of how EBV can affect the body, let's describe its cycle. The main idea to take from this slide is how EBV infects the host. The transmission begins with exchange of saliva. Hoagland, a virologist, concluded in 1955 that the most common transmission was oral, specifically kissing. The second type is via blood transfusion, but it's way less common. Droplets of the virus enter the mouth and quickly attaches and spreads to the tonsil ring, which is full of epithelial cells in the oral pharynx. Due to EBV's unique coding, its primary target are naive B lymphocytes. By attracting new, non-activated B cells, EBV has an easier time to infiltrate and progress quicker throughout the body. For that case, its incubation period is 4 to 7 weeks. To fully describe the highlighted key steps in the previous slide, I thought this illustration would be a great aid, especially for those visual learners. So we start off with the virus initially infecting the host via saliva. It then enters the epithelial cells in the oral pharynx, where replication begins. While replication begins, the virus infects the naive B cells. It starts to infect the underlying lymphoid tissue naive B cells and activates it with the growth transcription program. In this state, the viral envelope glycoproteins fuses with the host's cell membrane. This allows the cell to migrate into the follicle to initiate the germinal central reaction, seen right here. This establishes the default transcription program, also known as latency 2. From here, it provides survival signals to allow the cell to exit the germinal center into the memory B cell reservoir. When the virus enters the memory B cell reservoir, that's when we finish the primary infection. Persistent infection is when lytic replication becomes the highlight of the show. With lytic replication, memory B cells has the power to return to the tonsils. As stated before, the similar genes in lytic subdivision makes it easier for memory B cells to replicate and amplify the virus, leading to infecting other B cells or spreading to other hosts. It becomes more problematic when the resting memory B cells are sheeted from the immune system due to this. The illustration in front of us summarizes all of the key points and all of the details mentioned in the past 2 slides. To go over it one more time, we have the virus entering the host via saliva, infiltrating the epithelial cells in the oral pharynx, replicating and infiltrating B cells, becoming part of the B cell memory through latency, being part of the persistent infection, and making its way back to the epithelial cells in the tonsils or the oral pharynx to infiltrate other B cells. So why should we care? As previously mentioned, more than 90% of adults in the US has been exposed to this virus by the time they reach 30 years old. Fortunately for us, we have a good understanding of what is needed to be done in order to continue to control the infection. However, this was not the case for those before 1973. The virus went on for years before it was even considered an illness. It was first described in 1889, but it was overshadowed and treated as if it was the flu. Because many saw the virus was similar to the flu, many patients never fully recovered when given the proper treatment. More of an understanding of the virus was developed in 1920, where Spry and Evans accidentally noticed a discrepancy when noticing changes in blood levels, as they noticed there was a high increase in white blood cells. Then in 1964, EBV was discovered in a Burkitt lymphoma tumor, right here, through an electron microscope. This was then compared to the antibodies found in mono from 1932. Virologists linked EBV to mono in 1968, and it was then in 1973 where it was established as a disease, and we fully understood the signs and symptoms and the prevention and treatment. But what we didn't understand were those who were at higher risk to contract it. As previously stated, EBV is categorized as a human herpesvirus. With a name in the title, you would think it would be just humans. However, those that are more likely to contract the virus are humans and primates. Of the 100 plus types of herpes, there are only 8 human herpes, of which EBV is just one of them that we could contract. The biggest demographic that can contract the EBV are the adolescents and the young adults, the ages from 15 to 19. About 65 to 80% of this age group are more likely and at a higher risk at contracting. Another big factor to the contracting unit is a geographic location. This includes people that are away from the equator and live in industrialized countries. Other factors include race, as there is a higher chance for non-Hispanic blacks and Mexican Americans than there are non-Hispanic whites. Socioeconomic status, those who have more access to health insurance, lower household education, and lower income were statistically significant associated with higher antibody prevalence. Living situation and catchment area are similar as it relates to how crowded areas are around an individual. This can include daycares or living in the dorms. The signs and symptoms associated with mono never really show that it's actually mono. From this little photo right here, you can clearly see that mono affects all different types of systems throughout the whole body. It has a very generic type of symptoms, such as soreness, redness in the throat, lymph nodes swelling, respiratory cough, and all these other things, all similar to different illnesses such as the flu, the common cold, and so on and so forth. But there are different sides you could notice when someone does have mono. The first thing is an abrupt sore throat. So a sore throat that happens just out of nowhere. The sore throat lasts around 10 days. Along with the sore throat, you can notice that the person has a larger throat, meaning that it grew in size due to the lymph node enlargement. The second sign, one of the more important indicators, is myalgia or body aches and fatigue. This lasts around 20 days, and it's more of a prominent indication as it can have one person standing up out and about for one day and the next day be bedridden. The third sign, in which all hell breaks loose, is when a high fever develops. Usually during this time, many symptoms become more significant, such as rashes, a decrease in appetite, nausea, vomiting, and an enlarged spleen. Later on, we'll continue with how to treat all of these symptoms. But for right now, all these symptoms could last from 2 to 4 weeks. The signs and symptoms associated with mono are a pretty good indicator of a person having mono. However, it's not really confirmed until there's a diagnostic test. But the question is, what kind of diagnostic test would you run? The other white blood cells become more aware of when the virus enters the body and begins to replicate and infiltrate the B cells. Because of this, there's a high increase in white blood cell production. To diagnose mono, a simple blood test would be used. From this blood test, a white blood cell count would be conducted. A mono infection typically causes your body to produce more white blood cells as it tries to defend itself. Most patients with EBV have a mildly to moderately increased peripheral white blood cell count, usually in the range of 12 to 20,000 cells per microliter. After understanding how mono and EBV can affect the whole body from the cellular level to the systemic level, it should be taken with caution on how to prevent it and knowing its treatments. The most important and most obvious way of preventing EBV is taught from our friend Joey Tribbiani. Just don't share your food. In fact, just don't share anything that has any sort of droplets of saliva. Overall, just don't share anything. Along with that, make sure you wash your hands before you touch any of the orifices around your face, along with washing your hands before you eat. Just don't be gross. Unfortunately, there's no vaccine, so many of the treatment only relieves the symptoms. And of the main symptoms previously stated, the treatments are as followed: staying hydrated, getting plenty of rest, and taking over-the-counter medication for pain relief and the fever. Congratulations on making it all the way to the end of this presentation. If you'd like to know more about mono, in case you have it or just really curious, I recommend these sources. They're super informative and super detailed. Check them out if you really want to. Thank you for listening and watching.